Hi everyone, welcome to the Macromolecules Building Blocks of Life. This is the first of the lecture series. We're going to be discussing the synthesis and structure of DNA, proteins, carbohydrates, and fats. As always, remember that you need to revise the biochemistry section of the MCAT last because there's a lot of details and and there's only so much space in your brain to actually hold so much memory, So revise it last. So let's begin. DNA, proteins, carbohydrates, and fats are linear polymers, and they're made up of monomers. So that's the first fact that we need to know. Now let's start with the structure of DNA. So the DNA is a double-stranded molecule that are joined together by hydrogen bonds to form a double helix. Now the basic monomer is a nucleotide. What is a nucleotide made of? It's made of a negatively charged phosphate and sugar backbone added to a base. Now in DNA, there are 4 different types of bases, A, G, T, C, which stands for adenine, guanine, thymine, and cytosine. Now the discovery of the structure of DNA is not complete without mentioning the work of 2 very important scientists, one of them being Rosalind Franklin. As shown here, we have the results of her X-ray crystallography studies, which clearly shows the structure of the DNA, and the other being Erwin Chargaff, who discovered that the ratio of the bases were fixed, in the sense that the ratio of the base adenine in any given DNA molecule is always the same as the ratio of thymine, and similarly, the ratio of G is the same as C. So from this, Watson and Crick was able to deduce the double helical structure of the DNA. And the double strands were held together by hydrogen bonds, A being bonded to T with 2 hydrogen bonds, and G bonded to C with triple hydrogen bonds. It is necessary to distinguish between a DNA molecule and an RNA molecule. So the D in DNA stands for deoxyribonucleic. So as you can see here, 1 hydroxyl group has been removed from the ribose sugar backbone, and this is in contrast with the uracil base, which replaces thymine in RNA, where both the hydroxyl groups are still intact. The double-stranded DNA molecule is further coiled around a positively charged structural proteins called histone proteins. They're positively charged because they're rich in lysine and arginine residues, and they're attracted to the negatively charged phosphate backbone of the DNA. And the 8 proteins that make up the nucleosome cores include 2H2A, 2H2B, 2H3, and 2H4, and the only structural protein is H1, which does not make up part of the core. It attaches instead to the linker DNA. Now we come to the part of synthesis reactions. When it comes to synthesis reactions, it's not necessary to memorize every step of the pathway because I think it'll be too much. So what you need to do in every reaction is to remember the rate-limiting enzyme and the ingredients or substrates for the reactions to occur. So we'll start with the synthesis of the bases. In pyrimidine synthesis, the synthesis of C and T, the rate-limiting enzyme is carbonoyl phosphate synthase, abbreviated CPS2. Remember, is the number 2. And the ingredients or substrates necessary for this reaction is aspartate, carbonoyl phosphate, CO2 carbon dioxide, glutamine, and ATP. Now this reaction is important because there are several chemotherapy and antibiotic drugs that act on this pathway to stop the proliferation of bacterial cells and also cancer cells. So as seen here, trimethoprim is an antibiotic that inhibits dihydrofolate reductase, and methotrexate similarly. 5-fluorouracil is a chemodrug that inhibits thymidylate synthase. As for purine synthesis, the synthesis of A and Gs, the rate-limiting enzyme is glutamine PRPP amidotransferase, and the ingredients or substrates is glycine, glutamine, aspartate, THF or tetrahydrofolate, and CO2 or carbon dioxide. Another thing you have to remember about purine synthesis is that the base is synthesized on the sugar phosphate backbone, and this is in contrast to the pyrimidines, where the base is synthesized first before the sugar phosphate backbone is actually added. Now, the diagram on the right actually shows the recycling of purines, and this is an important reaction because deficiencies in enzymes in this pathway actually translates to diseases. And the one enzyme that you must, if you must remember one only, it's HGPRT, which catalyzes the recycling of the breakdown products of GMPs and AMPs, which is guanine and hypoxanthine, to its precursor forms. Right, and eventually guanine and hypoxanthines both will be broken down further to form xanthine, and there is a convergence of two pathways to this byproduct, and xanthine is further converted to uric acid and excreted. Right, so the enzyme that catalyzes this is xanthine oxidase, which is inhibited by allopurinol, which is a gout drug. Now, deficiency in HGPRT leads to development of a disease called Lesch-Nahen syndrome, which is characterized by gout, lip biting, and chorea, which is writhing-like movement of the body, and intellectual disability. And the treatment for this is obviously allopurinol. Now, following this is DNA replication. All you need to remember is the key players. We start first with the primosome, which is a complex of helicase and primase. So the helicase basically unwind the double-stranded DNA so that other key enzymes could actually access the middle part so as to be able to create a copy of the strands. And we have primase, which is part of the complex as well. They lay down primers for the DNA synthesis to occur. And for structural proteins, we need to remember single-stranded binding proteins, which helps to prevent the double strands that has been unwound by the helicase from reannealing, right? And for keeping them apart, and also topoisomerase, which prevents supercoiling in other parts of DNA, while this part of the DNA is being unwound, right? And so the one key enzyme that you need to remember for DNA replication is obviously the one that creates the copies of the strand, and this is DNA polymerase 3 in prokaryotes, right? And this polymerase moves in one direction only, and so that the DNA strands are always created from 5' to 3' end, remember that? And because there are two strands to be replicated at the same time, then you have a distinction between the leading strand and the lagging strand, because the leading strand is synthesized continuously as the DNA poly moves in one direction, and then the other strand, the lagging strand, has to be looped so that it can only be synthesized in fragments, right, of DNA, and these fragments are called Okazaki fragments. Okay, and eventually an enzyme called DNA ligase have to anneal or sort of ligate the fragments so that it can become one continuous strand of DNA. Now, the DNA replication has to be faithful because we want to prevent mutations from happening to the gene. And there are 4 different types of mutations that may or may not create problems. So the first one is missense mutation, where there's a single base that has been changed, and it may or may not translate to a difference in the protein produced. And the second one is a nonsense mutation. That is a mutation to one of the 3 stop codons. And the 3 stop codons are UAGs, UGA, and UAA. Later on during translation, we'll see how the ribosome will stop translating the proteins when they encounter one of these 3 stop codons. Now, the next mutation is frame shift mutation, where you basically have an insertion at a point in the DNA which causes a shift in the reading frame of everything that is downstream. So this causes a pretty major changes to the way the DNA is read. And the fourth one is the creation of pyrimidine dimers, and this is due to exposure to UV light. Now, you shouldn't worry too much about these DNA mutations happening in your body, because there's DNA repair mechanisms that correct these. The first one being mismatch repair, and this happens during DNA replication itself. So a defect in these DNA repair mechanisms can lead to cancers. So in this case is HNPCC, hereditary non-polyposis colorectal cancer. And the second DNA repair mechanism is nucleotide excision repair, where you excise the whole nucleotide that is wrongly copied. And defects in this repair mechanism can lead to xeroderma pigmentosum, which predisposes you to getting skin cancers such as SCC and BCC, squamous cell carcinoma and basal cell carcinoma. And then the third repair system is base excision repair, which you just remove the base and insert the correct one in. And the fourth one is non-homologous end joining in order to correct double-stranded DNA, right? And defects in this leads to a disease called ataxia telangiectasia. We've finally reached the end of our lecture, and next we'll discuss about protein structure and synthesis. So remember to do the quiz first to test your understanding of this chapter, okay? See you next.